So you can play this screencast after handing out the notes from the PowerPoint with the blanks that are for the students so that they can fill in the blanks as they go through the screencast or the PowerPoint if I happen to be there. And so we'll discuss each of these slides from my more complete PowerPoint so that they can get the spelling and so forth correct. Okay, so nucleic acids. This is all about nucleic acids and those that are used to make DNA. They are very abundant in high protein foods like steak and hamburger, and in sweet potatoes and bananas, in fruits like apples, in whole grain breads, and in seafood. What are nucleic acids? Well, they are the macromolecules that are used to construct deoxyribonucleic acid, or DNA, and that is the system in any organism that carries instructions for what that organism will be, and in many cases, what it will do. Sometimes your DNA controls you by using these codes that the backbone protects. It is the genetic material. These photographs here are micrographs of genes that are forming from the chromatin in the nucleus when we get into the reproductive cycle of DNA. DNA transfers information by making a template of RNA, and then proteins are made. We'll learn about that in the next section. When the chromatin, which by the way, your DNA molecules are 6 feet long in every one of your cells, and when the chromatin compresses at the beginning of DNA replication, which is a word used to describe the reproduction of DNA, DNA replication, the chromosomes are formed by packing the DNA strand into pieces and then packing them into ever tighter coils. Of the nucleic acids, there are RNA and there's DNA. The structure is made from nucleotides, which are a combination of a pentose sugar, a phosphate group, and a nitrogenous base. And those are the adenine, thymine, cytosine, guanine, or uracil in the case of RNA. One chromosome, number 1, in the human being is 246 million nucleotides long. This is a nucleotide. A nucleotide is the combination of the sugar, the phosphate group, which form the background, and a nitrogenous base, which connects to another base across the aisle in the helix, which makes up the DNA. There are 2 kinds of nucleotides. Whoops. There are purines, which are adenine and guanine, and there are pyrimidines, which are cytosine, thymine, and uracil. These nucleotides pair with each other and complement each other across the aisle in the DNA. Notice that the pyrimidines consist of 1 ring each, 1 carbon ring, and the purines consist of 2 carbon rings that are connected. The nucleic polymer is the backbone of DNA. On each side of the helix, there is a backbone of a phosphate group that's the phosphate group, a pentose sugar, ribose, and a nucleic acid, a purine and a pyrimidine. This is a fairly strong chemically bonded backbone where the chemical bonds are covalent, so they're strong bonds. But the nucleotides pair. Adenine always pairs with thymine in a DNA molecule. Guanine always pairs with cytosine. Notice that these two have 3 bonds to connect, and adenine and thymine only have 2 bonds in which they connect. That's what DNA looks like, and when we extract it from strawberries, you'll see something like that, but it'll be better than this small amount in this test tube. Okay. So DNA replication happens when these weaker hydrogen bonds between these bases of adenine, thymine, and guanine and cytosine break, and they separate, and this strand uncoils and opens up. In cell reproduction, DNA has to be copied before mitosis or meiosis can happen, because when the cell divides, each new cell in mitosis, which is reproduction where DNA is replicated and is exactly the same in the daughter cells, the DNA has to be copied during the latter part of the growth phase, which is interphase, and some people call the phase in which the DNA copies, is replicated, as pre-prophase. Watson and Crick were the 2 scientists working in England. One was actually from the United States, who discovered DNA in 1953. They didn't really discover DNA, by the way. They simply discovered what its structure, physical structure, actually was. They were in a race with Linus Pauling, this gentleman here, who everybody thought would find it first. He was a chemist from California. Yeah. And then there was another fella, Maurice Wilkins, and he had a partner. He was working with Rosalind Franklin. Her expertise was in X-ray crystallography. She took pictures of molecules with X-rays, and this is what DNA looked like. And it turns out that Watson and Crick figured out that that's a double helix, and that's how they discovered DNA. We'll watch a film about how this was done and about how amazing this discovery really was. Wow. Some people think that Rosalind Franklin should have been given a Nobel Prize. And so did Watson and Crick for her part in DNA, in discovering what the structure of DNA was. The problem is that she died from cancer before the Nobel Prize was awarded. And it turns out that the Nobel rules require that a person has to be alive to receive the Nobel Prize. So she didn't get it because she died of cancer. Very sad.